Oh, it says 2019, but actually this is 2020. I used this slide previously. So what I'm gonna, first, everybody hear me okay? Okay, yep, I see heads nodding, good. Okay. So we are going to look at the major proteins that exist in cells. And so our focus today is going to be on what's the function and the structure of DNA, the importance of RNA as an intermediate, and taking DNA information and converting it into protein. We'll look at what is a protein really and its function, which is largely to serve as a catalyst of enzymatic reactions. And this is done by the major class of proteins, which are called enzymes. So let's start though, I've got to click this here, by talking about scale. We're talking about the molecular world, and it's important to get some sense of really how small is small, as this slide shows. So if we think of things we can see easily, like a tennis ball, that's 10 times bigger than the period at the end of the sentence, and that's about 10 times bigger than a typical cell. So what this means is that the period that you see at the end of the sentence, if you were looking at some tissues, that's a space that's big enough to accommodate about 100 cells. All right. Now, this is the type of cell that would be found in plants and animals and visible cells, what we call eukaryotic cells, cells with nuclei. But if there are other major cells in the world, of course, are all the bacteria, and they're about 1/10 the size of a typical sort of human cell. And then today we're dealing with viruses, with the coronavirus, and the size of that is something like maybe 1/10 the size of a typical bacterial cell. All right, so the scale of cells here is then that you could take a typical virus like the coronavirus, and you could put 100 of them lined up across the typical cell. And a typical, you could take 100 of those to put across the period in a sentence. So these are big size differences. But when you start to look at molecules, you go down even smaller. So a protein, like an antibody protein, is, compared here, it's like 1,000th the size of a bacteria. In a sugar molecule, yeah, I said that wrong. That's 100th the size of a typical bacteria, the diameter. But a sugar molecule is like 1,000th. In other words, a typical bacterial cell, you could line up 1,000 sugar molecules to go from one side of the cell to the other. So this gives you some sense of size and scale. And we're going to be looking at molecules that are at the bottom end here, really small ones, things that are really difficult, or in some cases impossible to see with the best of all working microscopes. Okay, so let's look at a cell to begin with, to try and put this in perspective. We have two general categories of cells. First, we have the bacteria. And these two categories of cells I put here in the scale of how different they actually look in terms of their sizes. A typical bacteria in comparison to a typical cell in your skin, for example, they differ by about this much in size. Bacterial cells are much simpler in overall structure, which you can see, in this case, looking with an electron microscope. You really just see an outer wall and a membrane, and you see clear zones and dark zones, and these are the proteins involved in making other proteins. This is places in the cell where there's just soluble sugars and other small proteins, etc. When we look at a typical cell that we might find in our body, we find all this intracellular complexity. So we see this big nucleus here. That's where our DNA is kept. And this nucleus is surrounded by a double membrane. And then out here are all of these membranes here. They're involved in making other membranes. There are organelles involved in producing energy. There are organelles involved in recycling. There were organelles involved in transport. And at some point in the future, we may talk more about cell biology. But today, we're going to really look at the molecules. So if we had a powerful enough microscope, they only exist in science fiction, unfortunately, and we just zoomed in on this little piece here where there's a pore in this double membrane that allows this part of the cell to communicate with the nucleus here, it would look like this. Now, this is an attempt to show an accurate drawing of what the inside of a cell looks like, putting all of the molecules at their correct size. And so this here you see is the double membrane that surrounds the nucleus. And there's an opening in this double membrane, and it has this very complex set of proteins here that functions as a gate that allows some molecules to enter the nucleus, other molecules to exit the nucleus. And this flow from one compartment to the other is kind of organized, regulated by this complex here that sets in this opening. Now, what are all of these molecules? Most everything you see here are proteins. All these different colored molecules in here are different proteins. And in the molecular world, proteins are very big in that a typical protein could easily contain 10,000 to 100,000 atoms. Now, what we see here is the membranes, and they are made up of lipid molecules, which are kind of long linear molecules. Inside the nucleus here, this is where the DNA and RNA is. And in this drawing, the DNA is represented as blue. Whoops, let me go back here, hit the wrong button. So to give some sense of the scale of it then is inside the nucleus, if you could really see the molecules, a DNA molecule would be about this big, a very thin thread. And there are just so much of it here in actual size. I'm not saying an analogy here. I'm saying the actual size of the DNA in every nucleus in your cell is that this thread is about 6 feet long. So, you know, to try to get that in your head of the scale, the cell itself is so tiny, you know, just 1/100th the dimension of the period of sentence. And then inside that cell is the nucleus. And inside that nucleus is a thread that is 6 feet long. All right. And we're going to come back a little bit more to its structure. Now, let me say also that, you know, when I've given this talk before, it's always been to a live audience, and it could be more interactive. If somebody really has a burning question, don't be afraid to just unmute yourself and ask your question. So I'm very open to interruptions. In fact, is there anything up to this point? Somebody has some burning question. Okay, well, we'll move on then. All right, so let's look at what's called the central dogma of molecular biology. That is that the way life is made is to store the information for making a cell or an organism in a DNA molecule, this long thread-like molecule that we're going to talk about. We can think of this as the book of instructions in here. If the cell needs to make some particular protein, it goes into the book of instructions and makes a copy of one instruction set. If this book is like a cookbook, this would be a recipe for one particular thing in that book. And then this recipe is read by a protein machine that synthesizes a protein. And we're going to look at exactly what these different molecules are and how you get from DNA to RNA to protein. The central dogma of molecular biology. Okay, so here's a cartoon of what DNA looks like. And it's a very complex molecule. I'm going to show you more details. But It has a set of atoms that form a long linear structure. And in fact, you take two of these long linear structures and you put them side by side and connect them, and you get the helix, the DNA helix. This information in here is transcribed, which just means copied. You make a copy of the order of the rings on the ladder here into an RNA molecule, which is really a very close cousin of DNA in terms of the chemistry and the structure. And then this RNA molecule goes out into the cell, binds to an apparatus called the ribosome, and that's a manufacturing machine to make protein. And we'll talk about what that looks like too. A very different kind of molecule. These two are very similar, DNA and RNA. Protein is very different from them. All right, so what I'm going to do is I'm going to show you this cartoon, which gives you an
Here is a cell, the basic unit of...
I'm going to silence the sound on it here. Okay, and have to hit go. This is going to show you an overview of the whole thing. It's going to give you way too much information in a compact way, but it's an overview, and then in the end, we'll come back to it after we've described the details. So we're going inside the nucleus, and we see that the DNA in the cell sometimes is all compacted into chromosomes. Other times it's unraveled so that it can be accessed in red to be used to make proteins. So this is a way to represent the DNA, and a piece of the DNA is what makes a gene. Now here a protein is coming in to make a copy of a part of this DNA. It pulls the two strands apart, and the basic building blocks that make this new molecule comes in. These are hooked together to make a strand of RNA. And as you'll see, it looks very much like DNA. It has this long linear part with these side branches. But there are 4 different flavors of kind of rungs on this ladder, if you will, and that constitutes an alphabet, a code that give instructions to make protein. So that RNA then, after it's synthesized, goes out of the cell, out of the nucleus rather, out into the soupy part in the cell, and a big machine made of protein binds to it and basically reads the code. And what it does is, for 3 letters, that specifies bring in an amino acid to this synthesizing machine, the ribosome. All right, so if you had the letters U, C, and A down at this end, that might mean bring in alanine. And so it goes along this code 3 letters at a time, each 3 letters driving the transport into this machine of an amino acid. The amino acids that are then hooked up one by one to make this long chain. They go out a pore in the top of the ribosome. And you end up with a linear string of amino acids, but then they collapse and bind to each other. And in doing so, they make a protein. All right. So that was the whole central dogma. Whiz bang.
I have a question.
Yes.
Okay. It always, I never had this question answered, so maybe you can. What starts the process? What says to the DNA, it's time, I want this, or I want that? So do we know?
Yeah, I mean, it's somewhat a chicken and the egg kind of problem. But let's say that, take a simple case of a bacterium in your gut. If suddenly you've just eaten a meal and there's a lot of sucrose, sugar in that meal, that sugar will bind to the outside of the bacterial cell, and that'll trigger a reaction inside the cell saying, sugar is out here, bring it in. And that will bind to a protein inside the cell that will bind to the DNA in front of the gene that makes the protein to degrade the sugar.
I see. It
So
goes
it's a whole series of signaling pathways, which
okay.
are pretty well understood for many metabolic reactions. Okay. Okay, so let's go back to really the core of it. So this is one way to represent a DNA molecule in which you can see every atom in the DNA molecule. So its basic structure is what we have here is this long linear piece on the outside, and then attached to this linear piece are these sort of side branches. All right. These side branches here exist in 4 different structural forms: A, T, C, and G. You see they look somewhat similar. The major big differences is that A and G have 2 rings. There's a ring here and a ring here, 2 fused rings. Whereas T and C only have 1 ring. The structure is such that T will always stick to A's because this is slightly, there's an electrical positive and negative charge difference between these 2 molecules, and these weak electrical signals attract to each other and will allow A and T to bind to each other in this way. When C's and G's come together, they line up such that there are 3 places where a minus and a plus will attract each other and hold these together. These connections are very weak. And a really good analogy is to think of this as almost Velcro-like, in that if you have Velcro for just a few tiny connections, it takes very little effort to pull them apart. But if you have hundreds or thousands of connections, then these 2 strands in the DNA helix are actually held together quite tightly. All right. So to kind of back away from the atomic complexity of it, what we have is what's called the backbone. And the backbone is basically made up of sugar molecules and a type of molecule called a nitrate. Well, anyway, sugar molecules and phosphates. And then we have the nucleotides, the A, C, T, and G, that are attached to the backbone. So that a single strand of DNA, right, this one would be the backbone, and it would read T, G, C, and T, T. However, because A always binds with T, C always binds with G, this can be copied to put its complementary strand kind of on the other side here. So it has its backbone, and that will read in the complementary way. Where you have T, you have A, where you have G, you get C, where C, you get G. Now, I'm not going to go into it in this talk, but the core, the beauty of this is that if you pull these apart and you want to replicate it exactly, it's can be done because the whole code here is set up such that if you want to make a double helix from the single strand, you have to bring in an A molecule. For this one, you have to bring in another A molecule. For this one, you have to bring in a G molecule. In other words, you pull them apart, you can read it and replicate the whole strand. And one thing I'm planning to do with the course this year is, for my second lecture, I really want to return to many aspects of DNA that I'm just superficially going over today, and go into that in a little bit more depth. Now, this is what a DNA molecule would look like if you flattened it. Now, it's not really a planar structure. It's a very bumpy, lumpy structure. And so what it really does is it really forms this helical structure. And if, in fact, we looked at a model that represents every atom in the DNA, it really looks like this. So what you have here is the yellow is the phosphate that's in the backbone. Reds are oxygen molecules. Blues are nitrogens. Okay, whites are hydrogens. Grays in the middle are carbons, etc. Another thing to really emphasize, because the cartoon, or this model, which is an accurate model, really a kind of hard concept to grasp just looking at it, is that although this looks very uniform, it is not exactly the same here as it is here. And why? Because the sequence is different. So in this part here, you have a pair of T's, for example, TA base pairs. Down here, you don't have a pair of TA base pairs. That means the actual detailed structure of the molecule up here, the lumps and bumps on the outside, are different from the lumps and bumps down here. And that is the core to how DNA gets recognized. The difference in CT-A&G gives you a different surface structure in the DNA molecule. And this can be effectively read by protein molecules to find sequences in the DNA. It's really the heart of how the whole thing works. Okay, so let's go back then to look at DNA in the cell. And this picture here is really an attempt to show you how you take this 6 feet of DNA, this long, complicated molecule, and put it into a cell in a way that it isn't damaged and yet it's accessible. So when cells are dividing, the DNA is highly complex. It's wrapped up, supercoiled, we call this, into a big lumpy structure that we recognize as a chromosome. But when the cells are not dividing, it unravels so that this structure is exposed to the kind of the water out here in the nucleus. And these sequence differences can be sensed by proteins that are bouncing around here inside the nucleus. Okay. Now, in humans, when our cells are replicating, then we can see all of our DNA as chromosomes. And every human cell has 23 different kinds of chromosomes. And we get a set of 23 from our mother and a set of 23 from our father, giving us 46 altogether. For quite a few years, we've been able to use dyes to specifically label each of the different chromosomes. There are no, you know, I don't actually know how this is done. It's really quite a trick, but it can be technically done. And so if you want to, for example, in newborns, sometimes they will do what's called a karyotype analysis. Karyotype simply means, what do all your chromosomes look like? And what we're looking for here is some abnormality in a chromosome. All right. So the way this is actually done is you take some cells, you kind of squish them on a microscope slide, you add these stains so that the chromosomes become visible, and then in the past, they would actually kind of cut out the photograph. Now, of course, you can do this all on a computer. So you line up the 2 longest ones, the second longest ones, the third longest ones, and this is called chromosome 1, chromosome 2, chromosome 3, all right, through the whole set. Now, one thing you can just see is that is this male or female? Well, it's a female. You can tell that because there are 2 X's in here. If this were a male, it would only have one of these big chromosomes, and then it would have a stubby little Y chromosome, difference between males and females. And if you were doing an analysis here, you would get this odd finding that the smallest chromosome, the two are not exactly the same. And if you were missing this much of a chromosome, this cell would die. So how can you get that? Well, in this particular example, it turns out this chromosome was broken and the other piece of it stuck to chromosome number 9 up here. So this is an anomaly. Would that be good or bad? Most of the time it would be bad. But if in fact the break didn't actually break in the middle of a gene, this person could actually be rather phenotypically normal. All right. So anyway, you can see the DNA in your cells, but keep in mind that even this single chromosome, the DNA here, if you unraveled it, is probably about 5 inches long. It's exceedingly thin, but 5 inches long. Okay. Now, just a few other points here. So this is called our genome. Genome is just the word that means the collection of all of your genes and all of your DNA. In humans, we have 3 billion base pairs. That's 9 zeros. And within these 3 billion base pairs, there are regions that code for 25,000 genes, roughly. And that number is, over the years, we're kind of settling on that number. And this interesting fact here, that over 90% of our DNA is not in genes per se. It's
Can I ask a question?
Was that a question?
Yeah. When you're defining genes, are you talking about just ones that translate into proteins?
Not necessarily. No. So, for example, there are genes that encode the RNA that go into ribosomes, and those are considered genes. Gene is really defined as something that makes a product that kind of persists and is used in some kind of structure. But 99% of them are making proteins, and about 1% of them make RNA molecules. All right. So what you have is spacer DNA in between the genes, but the other thing you have, it's estimated about 15% of all the DNA is just old viruses, old kind of pieces of self-replicating DNA that infected our ancestors hundreds of thousands of years ago, and it just persists in our genome. All right. So in looking at this, non-gene DNA is actually a very active area of modern molecular biology. Okay, so actually, that's a good point. Any other questions at this point? Because we're going to slightly switch gears here on the kind of basic DNA structure. Okay, hearing none. So what is a gene? So I'm showing you here the coding sequence for a real gene. So what you see is a representation of what the base pairs, the rungs on the ladder of DNA, what they are for this particular piece of DNA, and it's reading just one strand of the double helix. So there's a convention to know what strand is being represented. So one is, if this is a gene, this would be the coding strand, the sequence of the coding strand of that DNA. And just looking at the basic alphabet code you have here, there's not obvious information. There's no punctuation marks. It's just continuous. Some of your chromosomes are, let's see, probably your biggest chromosomes are anyway, more than 10 million uninterrupted letters in a row. So how do we know where a gene starts and where one stops? And the answer is the information is in the sequence itself.
Hey, Barry?
Yes.
Who came up with those 4 letters? Like, why did they decide on A, C, T, and G?
Okay, it's because the names of these molecules are adenine, cytosine, thymine, and guanine. So it's kind of straightforward from the chemistry of what makes them up. And we'll see that the T, thymine, comes in a slightly different chemical form, which is called uracil. And the only difference between DNA and RNA are one key difference between your DNA and RNA is that in DNA you use T and in RNA you use U. We'll come to that in a bit. Okay, so we now have worked out what these sequences mean to some extent. And we know that certain sequences specify the kind of punctuation, stop and start signals within a gene. So you have small sequences often made up of only as few as 6 to maybe 12 nucleotides. And what these are sites that can be recognized by proteins as the starting place for a gene. So this particular piece here, and on your screen, you know, my screen right now, I see all this zoom menu is pulled down. Do you see that? Is it blocking the top of the? It's not. Okay, good. All right. So that's TG, TTC, TC, AGT. That is the signal for one particular type of protein to recognize this as the start of a gene. And so what will happen is in the long DNA strand, it'll just be bouncing along here, kind of bumping against this piece of DNA until the lumps and bumps in the protein exactly complement the lumps and bumps of this particular piece of DNA, and that protein will bind right to this point. It'll stick right here. And that protein has the function of marking the start of a gene, and it will essentially provide instructions for other proteins to come in and say, I want to make a copy of this gene. And so it will start right here, and I'm going to show you how this copy is made in a cartoon form. And it will go down to the end down here. And down at the end here, there will be some signal that's saying end of the gene. These are rather more weakly defined. Now, within the piece that gets made going from here down to here, there are 2 other important signals. So if we look in here, we see that in this first place where we see the sequence ATG, that means start. It means the start of a protein. And so the protein gets read all the way through until we get to the sequence TAA, and that means stop. All right. And then this is kind of this actually gets made into RNA, but it doesn't get made into protein. It's kind of a tail on the end of the sequence. We think one of its function, for example, is these molecules can get chewed off at the end, and we don't want to lose any of our gene that's in here. So it's kind of a protective little tail on the end of the sequence. So let's go next and look at this question of the genetic code, the A, T, C, and G. You know, working this out was just kind of one of the thrills in molecular biology when they got the sequence, the structure of DNA, and they saw that it was a simple code with only 4 letters in it. And the idea was, how in the world are we going to figure out and break the code? And it was kind of worked out sort of one word at a time. Really nifty story that maybe I'll be able to talk about a little bit when I give my expanded DNA lecture. But what we know is that, in fact, 3 consecutive letters make a word. And what a word is and the central dogma of molecular biology is it specifies an amino acid. And this is what proteins are made out of. All right. So ATG means start. It also specifies a particular amino acid, the one methionine. And that means that almost all proteins always start with a methionine. There turned out to be 3 different letter combinations that mean stop: TAA, TAG, or TGA. All right. If you work out the math here, if you've got 4 different letters in your alphabet, and all your words are 3 letters long, how many possible words are there? And the answer turns out to be 64. 64. All right. There are not 64 amino acids, there are 20. And so you have situations where like GGA might mean lysine, but GGG will also mean lysine. So there's some, we say, redundancy in the code. All right, but your basic situation is, here's the gene as specified in its nucleotide sequence. ATG means start, and it also means methionine. CGT is the second amino acid, and that turns out to be an arginine. And GCG turns out to be a cysteine. So you take the gene, you've made a copy of it in RNA, which is identical except instead of using T's, you use U's rather. And so AUG means methionine, CGU means arginine, UGC means cysteine. You keep going until you get to a stop codon, and that brings you to the end. So this is kind of the core of what the genetic code is. And questions on this. All right. In a way, it's amazingly simple alphabet. And it is the same in all organisms, from plants to animals to bacteria. There are virtually no exceptions to this use of the genetic code. Okay, so we've looked at what a gene is and the stop and start. And I mentioned that what you've got in the sequence is some sequence that says to a protein, bind to this site and make an RNA copy. So let's say here's the gene, here's the start site. The big enzyme has come in. The function of this enzyme is to thus pull the 2 strands of the DNA apart. This is going to be the strand that's going to get copied down here. So as it's pulled apart, a T is exposed, so it brings in the A base to match up with this T. This one here is an A, so a U base is brought in. Then these are connected, and then you bring in another T, and that one, T will bind an A, and that'll get connected. A G will bind the C, and that'll get connected. And so you'll end up with this copy of your DNA, of one of the strands of the DNA, and it'll be copied into this molecule RNA. So I said at the beginning that DNA and RNA are nearly identical in their chemical structure. The really only difference is that U's are used instead of T's in RNA. And there is another important but subtle chemical difference, in that is that in the backbone, there is one atom difference. DNA is deoxyribonucleic acid. The ribo refers to the fact that it's made out of, in part, ribose sugar. But it's deoxyribose, meaning deoxy, missing an oxygen. RNA, on the other hand, is just ribonucleic acid. It's not missing the oxygen. This simple plus or minus of this oxygen molecule turns out to have a really important difference in the chemistry and the reactivity of the molecule. Fundamentally, the RNA form is more easily degraded and is less stable, but the DNA form is more difficult to degrade and is very stable. And of course, we now know that DNA molecules can last for tens of thousands of years, whereas RNA it is just very easily degraded in the environment, or really very short lifetime in cells also. Okay, so we've seen what our genes are. We make a copy of a gene into an RNA, and then we go to the next step. The next step is that RNA alphabet code is somehow used to string together a whole sequence of amino acids. So let me first look at what are amino acids. You know, they're arguably, you know, maybe the most important molecules in our body, because this is what does the chemistry. This is what metabolizes our food. It's what drives our muscles. It's what signals our neurons. It's the chemistry that can take place in proteins, which are made out of these building blocks, amino acids. In virtually all kinds of organisms, we have the same basic set of 20 amino acids. And so I'm showing you all 20 here. The core of it is that you have an amino group. NH2 is an amino group, nitrogen and 2 hydrogens, tends to be positively charged, connected to a carbon. And then on the other side of that carbon, you have a carboxyl group. So the carboxyl group is carbon connected to 2 oxygens, tends to be negative. So this oxygen here can O- will readily and often bind H+, which it gets out of water. Water is H2O, and water can dissociate to make H+. So often this will have a hydrogen on it. If it loses that hydrogen, that's what acid is. You say something is very acidic, that simply means that it's got a lot of H+ in it. So what we've got here is an amino end and an acidic end, and that's why this is called an amino acid, Now, that's the core in all of these molecules, amino carbon acid group, amino carbon acid group. All right. What they have on this central carbon, though, then is what we call the side chain. It can be as simple as just a hydrogen atom. It can be slightly more complex, having a carbon combined to 2 hydrogen atoms. Or it can have 3 of those complexes, like this, or it can have 4 of them, etc. In some cases, it can bind to a more complex ring-like molecule. In some cases, we said that this is an acidic group here. It can have another acidic group down at the end. These are examples of where we have an amino group here. We have another amino group down here at the end. What all of these differences specify really are 2 important chemical properties. One is how well do they bind water? Are they soluble or are they insoluble, basically? If they don't bind water, like these up here, all the ones in green, what they'll end up binding is really, they'll bind each other. They'll be excluded from the water. They'll be kind of fatty, we would say. On the other hand, these molecules down here all bind water rather readily, and they'll tend to stick out into the water. These down here have the important additional feature that they're charged. So these have negative charges on them, and these have positive charges on them. Negative is going to bind positive very strongly. So what you got is many ways in which these side chains can interact, can be attracted or repelled. And that's what drives the structure of a protein in the end. Okay, so this is the same 20 amino acids. It's just shown in a different way. It shows you that the simplest side chains have kind of no bump on the molecule. If you add a few atoms, you get a little bump. If you add more atoms, you get a bigger bump, big bumps like this. And so if we look at the kind of overall surface of these 20 different amino acids, they've all got the same core, but they all have different sizes, lumps and bumps. Some of these will bind water, some will not, some are positively charged. These are the basic modules that we use, these 20 amino acids that we use to make all kinds of proteins. Just one last thing to say about this, that in our diet, you know, this being the central kind of molecule that we need to make everything else, our bodies can only synthesize half of these. We have to get the others from our food. So plants, bacteria, they can make all 20. They have to make all 20. Plants don't eat other plants or animals. They've got to make everything for ourselves. So the reason, you know, that we need a balanced diet is to make sure that we get a diet that's going to provide us with all of the amino acids, especially the 20 that we cannot synthesize ourselves. Okay, so let's look a little bit then at how we go the last step in the central dogma, which is from RNA to protein. So we saw that fly by in the movie that I showed at the beginning here. But now let's look at this in a little more casual way here. The machine in the cell that drives the synthesis of protein is called the ribosome. It is basically a very large complex of proteins and RNA, it turns out. In our cells, I think our ribosomes have 80 different kinds of small proteins and 4 different kinds of small RNAs. And it makes this machine that has, boy, I don't remember, it's like a million different atoms. And one of the prides of our campus, UCSC, is probably arguably the world leader in understanding the structure and function of this ribosome is Harry Noller up at UCSC. And he is one of the people in the world who has figured out the structure of the exact position of all million atoms in this ribosome. It's a great feat. And he is now working on showing exactly how it does its job. But here's the core of it. So the ribosome can bind to an RNA molecule. This is it copied a particular gene. All right. And it's not shown in my cartoon, but it will bind it at one end where that ATG was located. It can recognize that as the start, and it will position that ATG at the right place in the ribosome. Then we have these special carrier molecules that are going to pair up the right amino acid with the correct 3-letter code. These are called tRNA molecules. They are a specialized form of RNA. The reason they need to be made of RNA is because the A's and the G's can recognize whether they're TTC. And any place in this code down here where there was TTC, in RNA, however, T is always U, so it would be UUC, and here's a UUC here. This will kind of effectively pair up, bind weakly to this 3-letter code, position it on the ribosome, bring in this amino acid and position it right next to this one, and the ribosome will then link the two together. All right, hook them up. And so then the ribosome will move the RNA along by 3 letters. The next tRNA will come in, bringing the correct amino acid. The amino acid will get hooked up, and then the whole process will repeat. And so in this way, this every 3-letter set here is going to bring in the appropriate amino acid. It's going to get hooked up, and you're going to be making this this chain of amino acids out at the other end here. Okay, any questions on this part here? What I'm going to show you now is of a movie, and these animations are getting better and better in terms of being not just cartoons, but really trying to draw all the structures to their actual size. And it's going to show you protein synthesis at the actual rate, actual speed that it occurs in the cell. And it's pretty darn fast. Okay, so here we go. Let me see, get my cursor here. All right. So, central dogma.
The DNA double helix contains 2 linear sequences.
All right. So, here's the DNA. Protein binds to the start site. That brings in the enzyme that copies the DNA and all its factors that it's needed. Once everything is assembled, you say go. And so it zips along. This is the real speed. It really just flies along, and it's making the RNA copy of that gene. And a typical gene is often about 3,000 nucleotides long. So that will often take about a minute. It does quite a few nucleotides per second. So what we've got here now is the RNA copy of that particular strand of DNA, that particular gene. Now, there's a real odd complexity that comes involved in here, and it turns out that some of this RNA can't actually be included in the final protein. It's called intervening sequence, and it has to be edited and cut out. And so these proteins are recognizing the green part as a part that has to be edited out before we move on. In my digging deeper into DNA, I'll come back to what this is all about. This is called a gene splicing. Doesn't actually splice the gene, it splices the RNA that was copied from that gene. So the parts that need to be cut out are cut out by the splicing machinery. Splicing machinery is made of protein. And so you finally end up with the actual coding part. And that goes out one of these pores in the nucleus. And there it encounters a ribosome, the protein manufacturing machine. And once the ribosome comes in, then it can position the RNA in the right place. And these carrier molecules, the tRNAs, will bring in the correct amino acid that corresponds to the 3-letter code. And it flies along, and it occurs at the rate of about 100 per second, something like this. And so a typical protein will have, maybe at least 500 amino acids in it. So it still takes a while. It still takes a minute or two to make a protein. And so the protein comes out at the top of the ribosome, and it's released, and it folds up into its final form. So that is the process of translation. We're translating the DNA code into a different sequence, into an amino acid sequence. So let's look then at this final step, which is the production of the protein and the function of the protein. So in this cartoon, just want to illustrate the fact that this strand of amino acids will have some that, for example, in some cases, one of them might be positively charged and the other is negatively charged, so they will stick to each other. In other cases, they might not be charged or compatible at all, and they might actually repel one another. And it may be that an amino acid at the end of the chain ends up binding most tightly to something that's way back in the middle of the chain. And so it starts out as just a long chain and it kind of flops around until you've got the sort of maximum binding sites that can be achieved. This is called protein folding. It's one of the big challenges of molecular biology to figure out how it gets the right amino acids stuck to its corresponding amino acids somewhere else in the chain. This is, as you can see, an immensely difficult calculation, just how it achieves its final structure. But it does, and it does it quickly, and in most cases, it does it completely on its own. In other words, there's no apparatus that's required to wrap up this chain into the correct final structure. All right. Sort of 95% plus of all proteins function as enzymes. And so this is just two ways to portray an enzyme. So at its core, it's a strand of amino acids. And so it's often shown in this form, which we call a ribbon diagram. And this comes out of the fact that there are some sequences of amino acids that form a fairly regular structure. They can be kind of like helical. In other places, they're just sort of random. In other places, they're sort of like flattened sheets that lie close to each other. And so we can represent these sort of regular folding patterns with this sort of regular diagram. They can be held together by many different kinds of interactions. I mentioned plus to minus, et cetera, or lipid-like things to other lipid-like things. One that is a really strong bond is there's one amino acid, cysteine, contains a sulfur in it, and actually forms a strong molecular bond. So if there's a cysteine here and a cysteine here, they will form a strong molecular bond. And in this particular example, there are 4 of these, what we call disulfides, that help to hold this whole thing together. Now, if we wanted to look at where are all the atoms in this chain of amino acids, then we can show an atomic structure like here. You can see there are pluses and minuses to both ways of representing us. This actually allows us to follow the chain. Here we can't really see what the chain is, but this allows us to get a good look at what the actual surface of the protein is like. And if we looked at this one, for example, there's a big cavity in here, and this may well be where the chemistry occurs. Let's say that this particular enzyme, in fact, is involved in cutting up RNA molecules. And it could be that an RNA molecule lies across this, that side chain sticks into this cavity, and that may allow the chemical reaction to occur to slice it in two. All right. So just to elaborate on this point a bit more. So here's a protein. It's folded up into its final form. And let me emphasize, this is not random in any way. This is a stable, particular structure that it attains. And in this particular protein, there's this big cavity here. And it is just the right size and shape, kind of lock and key like, to bind to some substrate molecule. this could be, this could be some complex sugar, for example. And once it's locked into here, the fact that you have all of these chemical interactions on the surface, and it can interact with other things here, it can either split this into pieces, or it could add another molecule to make it bigger, so it can degrade it, it can build it. But the point is that it promotes chemistry, and it promotes it for very specific molecules. So we have 25,000 genes. 95% of those are making 25,000 different kinds of proteins, which can bind to 25,000 different kinds of small molecules, all the amino acids, vitamins, nucleotides, sugars, lipids, etc., that are required to make a cell. And some of these are degrading, some of these are synthesizing, kind of all the set of what we need to make all of the chemical reactions to make a cell grow. Okay. Now, just a couple of final points. One is, what makes this all go? How does this substrate, the substrate is the chemical that's going to be changed in this reaction. How does it find this pocket here? What actually makes the whole thing go? And the answer is that it is just random collisions. So this is in water. And if this were a sugar molecule, for example, you will have millions of them banging around in the water. And they're going to bounce off of this enzyme. But every once in a while, bouncing randomly, one is going to bang into this pocket, and it's going to have a size and shape that makes it fit precisely in there, and it will stick. And it will allow the chemistry to occur. So things that are at moderate levels in our cells, are sort of millimolar concentration in the cell. This might be like glucose in your cells. They will encounter an enzyme just by this random collection. The chance that a sugar molecule at this concentration will hit any particular enzyme to which it has a binding site, that chance is that it will occur 1,000,000 times every second. So enzymes and substrates don't have to find each other. They're just banging around, kind of doing this testing. Do I fit here? Do I fit here? Do I fit here? At super high rates, kind of higher rates than we can conceive of. And at our body temperature, if it's at a typical concentration, this is going to happen 1,000,000 times per second, which means that in principle, this enzyme could do 1,000,000 chemical reactions in a second. And there are enzymes that work almost that fast. It's called catalytic perfection. One example is that the water molecules in your cell every once in a while make peroxide, hydrogen peroxide. That's a really toxic chemical, bleaches your hair. It reacts. You don't want a lot of peroxide. So you have enzymes just sitting there waiting for a hydrogen peroxide molecule to bump into each of them. And as soon as that does, they combine it with water and basically destroy the peroxide, make it go back to water again eventually. And those enzymes work near to catalytic perfection. They can catalyze reactions at up to 1,000,000 per second at this concentration. Okay, so to give a kind of sense of this, this is a cartoon that was done on a supercomputer to look at a molecular reaction with real size to see what really happens. So in this case, this is the protein back here. And they've kind of split it in two. So we're looking at the inside of a protein. This protein normally sits in a membrane. So it's in a barrier here. And its function is to allow water molecules. See, there's H2O, 2 hydrogens and an oxygen. That's water, molecule of water. It allows water molecules to move in from one side of a membrane to another by going through, there's a cavity in the protein here, and then there's a couple of amino acids that form a gate right in the middle, and they take a water molecule and allow it to pass through here and go inside the cell. You know, you want to keep the good things in and the bad things out. So you need a hole that'll allow water to go through, but you don't want salt to go through. You don't want sugars to go through. You just want water. And so it has this gate that is just the right size and structure for only water molecules. So how long does it take a water molecule to go through? Well, this is the reactions that occur in, I think this is like a picosecond of time, and they've colored one of the water molecules yellow here, so you can see it. So this is just like a millionth of a second watching a water molecule go through. And it makes this point that it's just all random collisions. It's all bouncing around in here. You can see our cavity, and here is our yellow one. It's kind of pushing its way through. It gets to the key gate part here, and it binds to this amino acid, and then that amino acid allows it to kind of bind here, and then by kind of random collision, it sort of works its way into the out part, but what? It went back. But then finally it goes down again and goes back, but eventually it gets water molecules in between it and it escapes to the other side. So, you know, the beauty of this is that as complex and as sophisticated as all of this chemistry is, it's ultimately just driven by chaos, just driven by this random collision. It's something that's rather difficult to get your head around. Okay, so now that you've seen all of the details, let's take another look
Here is a cell, the basic unit
at the
of
big picture here. So we saw that, okay, we're gonna zoom into our cell. So we've got the outer membrane of the cell. We've got all of the various organelles, and then we zip into the nucleus of the cell through one of these pores in the membrane. In there, we have the DNA, which is sometimes compressed into chromosomes, but more frequently is unraveled so that the proteins can be bouncing along there looking for binding sites that indicate the start of genes. That if the proteins are available to mark the start of genes, and that might be signaled by something you ate outside your cell, it will bring in the enzyme that will make a copy of a gene, transcribe it into an RNA molecule. So the 4-letter code will be assembled in the new RNA. And that RNA will then leave the nucleus. Okay, this is the messenger RNA and encodes for a protein, a sequence of amino acids. Okay, I told you about this splicing reaction that has to occur for most of our genes anyway. Goes out of the nucleus, encounters the ribosome, which is the machine that can read this code, assembles on the ribosome, then these tRNA molecules, the carriers of the amino acids, come in, and they do the translation process. They say, this amino acid should be inserted wherever I see the code AAT, for example. And so it will bring the right amino acid to the right place. And then the ribosome will attach those amino acids that are brought in to make this chain, this molecular chain. That's actually fed through a channel that exists in the ribosome, at the top of the ribosome. And the chain of amino acids, 20 different kinds of amino acids, but in a specific sequence, they will interact and bind to each other until they fold up into a protein with a very specific shape. And on the surface of this protein are binding sites for other chemicals in the cell. And that's what drives all of the chemistry of life. So we go back here to where we started with, looking at all of this molecular complexity. Here's inside the nucleus, the DNA getting transcribed into RNA, the RNA going out here, the RNA binding the ribosomes. These are the ribosomes making new proteins, and you kind of fill the cell with all of these different kinds of proteins. Okay, so let me just mention this, and then I'll open this up to questions. So the course, there are going to be 3 more lectures. At the end, I'm going to kind of digging deeper into DNA. I mean, how is it replicated? What's the stuff that's not genes? How big are genes, and what's this splicing all about? Maybe tell you a little bit about, how do all these new DNA sequencing technologies work that, allow us to, go out and collect bird poop in the woods and be able to identify individual birds, not just species of birds, but individual birds. How in the world can we do this with DNA, et cetera? Lots of interesting DNA things to cover. Our next speaker is going to be Josh Arribere. He's a rather new, young assistant professor. I think he's in his 2nd or 3rd year here. Came from Stanford. In fact, he was in a Nobel Prize winner's lab at Stanford. And he is looking at the question of how do cells maintain quality control? Because, if I'm saying so much of this is done by random collisions, aren't there a lot of mistakes made every once in a while? And the answer is yes, there are mistakes made. But cells have figured out ways to detect good proteins from bad proteins, good message from bad message. And how do they do this? Josh, in his experimental methods, is also heavily relying on the new CRISPR technology, and he may be able to tell us a little bit about CRISPR and gene editing. And then in this era of COVID, I thought, I've got to get Martha to talk to us. Martha has talked to this group a couple years back. She is an immunologist. And so one of the most knowledgeable people on campus is to understand exactly how our immune system deals with virus infections. And then the fact that many of the most serious cases of COVID infection, the serious diseases and the deaths, are often caused not by damage to the virus itself, but by our immune system overreacting. And she's going to talk about the pathology that comes about overreaction of the immune system. So those should all be interesting. So I hope I will see all of you for the final 3 lectures. All right, so with that, I open this to questions. Yeah, just unmute yourself if you have a question.
I have one.
Sure.
I'm just wondering about the location of the spliceosome. Is that in the nucleus, and then before the RNA comes out, it's all done in the nucleus? Or?
It is. It's all within the nucleus, and it's a really, for all I've shown these very complex pathways, transcription, translation, et cetera. Removing those introns is even more complex and involves more proteins than any of the others. It's a bizarre thing, and we don't fully understand to this day why it happens. I'll talk a little bit about that in my digging deeper into DNA.
Raise your hand.
Okay, Linda and Gordon.
Hi, I was wondering where the tRNA comes from that the ribosomes are busy working with?
So in the DNA, some of the genes encode DNA that specifies the sequence of nucleotides in the tRNA. So in fact, there are a couple hundred genes in our bodies that encode different tRNAs. So, you know, you've got 64 codons that have to be read. It turns out that you don't make the full set of 64 different tRNAs, but we make about 40 different kinds. But they're genes, just like protein codons. Yeah. Okay, other questions?
I have a question. This is Kitty. I don't know. You can't see me, I think.
Yeah,
but,
go ahead.
yeah. So did you say that 90% of the genetic material of the DNA is not genes? It's a lot of it's old viruses that invaded our bodies?
That is correct. It's
okay. I just want to make sure you got that number right. That's pretty big.
Yeah. I mean, originally a lot of this was called junk DNA, and a lot of it we think still is junk DNA. However, one of the big things that's happened in the last probably going on 10 years now is that we know that a lot of the DNA is converted to RNA even though it doesn't code for a gene. Some of that just gets degraded. It's as though the machinery, the copying machinery is just making a lot of mistakes, but other parts of it function to regulate which genes are turned on and which genes are turned off. You know, we really kind of understand a lot of the core processes pretty well, but the really challenging thing is what's on and what's off, and is it on a lot or on a little? You know, you think of this challenge, you've got 25,000 different kinds of genes. You know, the genes to make red blood cells and muscles, those genes are present in the cells in your brain. But you don't want them making muscles and red blood cells when they're supposed to be making neurons. And so they've got to turn on just the right set of genes at the right time and the right amount. And some of this junk DNA turns out to be involved in regulating what gets turned on when and how much.
Okay, so we don't really know like the percentage of that that's not old viruses that attacked us.
Yeah, but it kind of, you know, about 5% is genes, about 15% is viruses, and 80% of it is something else.
Okay, great. Yeah. And then just I could just look this up, but just for clarity on the amino acids. So how many are not reproducible by humans?
10, basically.
10 of the 20?
Yeah.
Okay.
Yeah.
Okay, thanks.
I'd like to ask another one. There's a remnant of a gene that used to be able to make, in earlier animals could make vitamin C. And I think it's in all the primates that have the same defect in the vitamin C synthesizing. So I think, I don't know how many there are, maybe you do, just genes that are no longer functional, that are still there, but don't serve a purpose anymore.
There are a lot. They call them pseudogenes because they have start and stop signals. And sometimes they have, some of them are made into RNA and then it just gets degraded, often because it makes a nonsense protein. A typical thing is a pseudogene. If you compare the sequence, it will look like, you know, 98% identical to a functional gene in another organism. But in humans, there have been stop codons, TAAs, TAGs, inserted in there, so it won't actually make a functional protein. But there are a lot of those. Lois, you had a question?
I have a comment. what I think, you know, this is also fascinating, but I'm sure what is fascinating is how people, men and women, have figured this all out.
Mhm.
I mentioned that I would love a course on the history of molecular biology. I think it would be so interesting. We all read that early book on, you know, finding the helix or whatever.
The double helix, probably, yeah. Jim Watson, yeah.
But there's so much more, and I think it would be really fun and interesting, you know, what's happened at UCSC, but what happened,
Mm-hmm.
you know, many years ago.
Yeah.
You know,
well, I like that idea, and it's something I've been interested in a long time, and I have read a lot of that. In fact, I thought at one point that would be another angle on that is going through science biographies. I mean, just some really fascinating things out there. Yeah, we should think about that. That's a good idea. Okay.
Don't other people hear what you like to hear the history of how it all happened?
Yeah. Louise, you had your hand up.
Yeah, I want to agree with that and supplement what she just said. I think what would be interesting as a part of the history is how scientists model what they cannot see. How do they work with these things that cannot even be seen, and yet they find evidence that teaches them how to draw models that would try to explain relationships between all of these unseen things. If tied into the history, it's part of the scientific method, and I'd love to see that.
Okay, I like that idea too. Okay, Louise, you had a question.
Yeah, I think the idea of your teaching more is a real fun one. I'll sign up. But my question, when you talk about things that, the parts that don't have a purpose, I think about the body parts we used to think didn't have a purpose, but it's just we didn't know it. And I'm wondering what you think might be the percentage of things that actually have a purpose we just don't know.
You mean of this 80%, et cetera? Well, we slowly are figuring out that some of that stuff we thought was junk does have a role, but I would say most of it is still continuing to be mysterious, and it is, you know, a dramatic result of evolution is we carry so much of our old history with us, in not just the successful parts, but a lot of the garbage we carry with us too at a molecular level. Interesting problem. Yeah. Let's see, Constantine. You're muted, Constantine.
Yeah, right. So to add more complexity to all this, I've read some research that scientists have been dabbing into adding more letters to the chain. Are you familiar with some of that research that's actually being done?
A little bit. Yeah. In fact, I think you sent me some links.
Okay, yeah.
Yeah. So it's interesting. So in principle, they're saying there's no reason why we couldn't add some new base pairs in a kind of an artificial system, and it does work. And it might allow kind of the synthesis of products that are not made naturally. So you could because as good as organic chemists are, there are still lots of molecules that are just impossible or impractical to make in the lab, but organisms can make them. Jack.
I have a question. You've presented these with the connection. So I see a gene, but we're each of us different, and our genes are different in some way. Could you indicate where the different why one is different from another, why I'm different from you. Where does that show up?
Okay. All right. Well, it's you know, it's fairly complicated at, you know, if we went into the details, but the simple overview is that we all have the kind of same core set in the same order, and the actual sequence of nucleotides is just like 99.9% identical. All right, but there are variants. So, you know, this gets you back to this sort of basic, you know, Gregor Mendel and genetics, in that you have tall plants and short plants. And if you look at the details, this is often due to just a single change, one A nucleotide in 3,000 that can exist as an A in one organism and a G in another. And that will affect one amino acid in the protein. And that may make the protein work a little better or a little worse. And so you'll make a little more of a product or a little less of a product. We call those alleles. They're variants of a given gene. And so there are these mutations that occur out in there. That, in fact, is the basis of, you know, all of this genetic ancestry mapping of your DNA. What they're looking for there, of course, are the differences, but they're really subtle. They're kind of like 1 in 1,000 base sequences. And if you make a change in the DNA, most of the time it will be neutral. And some of the time it'll make a change, but not necessarily a bad one. It just make a little more or less of something. And that's where you get all this difference. Now, then on top of that, you have the thing that, like a simple difference of hair color is not determined by one gene. So you might have a gene that says, make red pigment, don't make red pigment. Make black pigment, don't make black pigment. Make high levels, make low levels. So you could have 5 different genes that affect it. And so you could really see it as like having a palette of different colors of paint. And with just 5 variations, you can make 50 different tones. And so that's the other way we vary. So fundamentally, it's that for any given gene, there are many different minor variants. And then for any given character, there are multiple genes that are involved. And so it's a kind of exponential opportunities for complexity to occur. Okay. Oh, Marjorie. You're muted, Marjorie. Yeah.
Okay. Unmuted.
Yes.
I was given a supplement of, oh dear, L-lysine when I had shingles to stop its movement. What is lysine? What kind of amino acid, and what does it do?
So it's one of the 20. Lysine is an important one. They're all important, of course, but it has a charge. There's a plus charge on it. So it's an example of, if you change the code for a lysine, you're more likely to really affect the shape of a protein than if you change the code for another end. But as far as what it does, there are so many possibilities that you're looking at something that is in every protein in your body, and so it's not going to be at this genetic level. It's somehow going to affect the the metabolism of something that uses lysine. I don't know. I wonder if they know, in fact, or if this is just experience that it has the effect. Yeah. Oh, that's a really complicated one. Okay, Pat, and you're muted also. And okay. Okay. Didn't have a question. All right. All right. I think we've finished out. All right. Time for dancing in the streets, folks. Get out there. All right. So we'll see you all next week, and it'll be Josh Arribere, and he's a Sharpie, so it'll be good. All right.
Thank you, Barry.
Okay.
Barry.